Welcome to the Digital Cloud Forum series, Genomics in the Cloud. My name is Luis Salcedo, and I'm a cloud advisor at Google. I would like to introduce you to our esteemed speakers, starting with Geraldine van der Wera. She is the Director of Outreach and Communications for the Data Sciences Platform at the Broad Institute. She has an extensive research, biotech, and microbiology background, and she speaks three languages. She is a co author of Genomics in the Cloud, a book that was just published in April. Brian O'Connor is a principal investigator at the Broad Institute and has been leading genomics work in the cloud for over 10 years. He has supported biotech research and worldwide academic projects. He co authored the Genomics in the Cloud book with Geraldine. Finally, Joe Cronin is a Google Cloud customer engineer, and he works with Geraldine, Brian, and our friends at the Broad Institute. He will be our host for today's webinar, and he has been helping deliver engineering solutions to customers for a few decades, bringing a vast customer engineering experience to Google, where he has been serving for over 8 years. He was previously at HP Autonomy for almost 20 years. We are lucky to have these three individuals share some knowledge with us today. With that, I will turn it over to Joe.
Thank you, Luis. Once again, as Luis said, my name is Joe Cronin. I've been with Google for about 8 years and about 6 of them on Google Cloud. For the last 4.5 years, I've been supporting the Broad. The Broad's journey to the cloud started about 6 years ago. They were like many other companies who were playing the game of constantly upgrading their on-premise cluster to meet the demands of their researchers. But it became clear that they were just not going to be able to keep up with the dynamics as they needed to be. By moving to the cloud, they no longer have to guess about the projections of disk space that they need for the next 3 years. Google's engineering team worked with the Broad to create the Life Sciences API, which makes it possible to use Broad tools like Cromwell and GATK in the cloud, which you will hear about more today. With that, Geraldine and Brian, welcome and thanks for joining us today.
Thanks, Joe. It's really a pleasure to be here. Brian and I are both with the Data Sciences Platform at the Broad Institute, which is a part of the organization that is focused on trying to accelerate science, transform medicine, and improve lives through data technologies. And so we're part of the group that is developing infrastructure and operating services and software that aims to help researchers get their work done. And as Joe mentioned, we started moving to the cloud about 5 to 6 years ago. And I've been with the Broad for about 8 years. And for the first half of that, I would say we were doing all of our data processing for the Broad Genomics sequencing facility on an on-prem high-performance computing cluster. And as he mentioned, we realized that we weren't going to be able to keep up in terms of processing. You can see in the graph here that shows the increase in the amount of data that we've been generating just in terms of genome and exome sequencing over the past decade or so, that we've had a dramatic increase in the amount of data generated. And at this point, we're sequencing a new whole genome about every 5 minutes, and that's a huge amount of data. So when we moved to the cloud initially, it was really about being able to keep up with the processing needs of our sequencing facility. But beyond that, there's actually just the question of how do we deal with all that data once it's been processed? Because it's also not just us. There are many organizations worldwide that are now generating huge data sets too. And these data have more value if they're taken together as opposed to separately. And so the big question is how do we share that data effectively to make it widely useful? And cloud is not the answer to everything, but it's definitely key to solving these problems. Because if you think about the traditional approach for how we share data, there's like a central location where the data is deposited, and then every researcher or research organization, needs to go in and download a copy of the data to their local systems to work on it. And there's many problems with that. I'm not going to go through the full laundry list, but one of the problems is that it's not sustainable. As we increase the size of these data sets that researchers need to use, most organizations don't have the ability to maintain full copies of all these data sets. And so where cloud really helps us enormously is in allowing us to have the data stored in a place that's co-located with computing infrastructure. So instead of bringing the data to the researchers, we bring the researchers to the data. And so there's computing facility that exists in proximity to the data, so everybody can operate on the same, read from the same master copy, do the work. There's several advantages to that in addition to just the sustainability angle. It makes it a lot easier to share tools because you're working in shareable computing environments. And it's also a lot easier to provide auditable security systems and access controls so that we can secure data that is access controlled and protected, since we're talking in many cases about patient data, about human data that we need to protect. And so this is really the idea. The central idea is to bring researchers to the data on a public cloud system. And the goal ultimately is to really enable the research community to run federated data analyses. And that means, let's say you have a couple of data repositories with data sets that are particularly interesting if they're taken together, but they're hosted and owned by different organizations. Well, if they're hosted on the cloud, we can actually access data from both repositories, access tools that are in a tool repository that's also accessible from the cloud, bring all of that together in a workspace, and compute on these data in place without having to download anything anywhere. And so this is a very, very powerful idea that we're participating in building this ecosystem, which amounts to kind of a data biosphere in which people can participate and come to analyze the data much more conveniently than what is possible with the kind of old model of data sharing. So that's what we're driving towards, what we're trying to enable. And I will say this is applicable to pretty much all domains within life sciences. It's not specific to genomics. And we're actually involved in projects that touch many different kinds of life sciences data types and types of investigation, including single cell transcriptomics, including metabolomics, proteomics, but there's also clinical data, phenotype data, all of that. You can bring that together into the data biospace. So that's what we're trying to do. Now, we promised you a webinar on genomics specifically, so let's see how this applies in the context of genomics. Let's talk about genomics in the cloud. Now, for those of you who are coming from perhaps a different field, I'm talking about genomics as the science of human genomic variation. So if you take, random human genome, you've got about 3 gigabases of information encoded in your DNA. With high throughput sequencing techniques, we can read that information out into a huge pile of hundreds gigabase file that's composed of very short sequences. Then we analyze that huge pile of information, and for any given genome, we expect to see 4 to 5 million small differences compared to a kind of a standardized reference that stands as a standard human being, which is a complicated concept that's worth going into at another time. But let's say, okay, we have these 4 to 5 million small differences. And now the next step is going to be finding out which differences matter. So really, in summary, you've got this huge haystack, and you're not looking for one needle. You have 4 to 5 million needles in the haystack. And then you still need to filter that down to figure out which needles are actually relevant to what you're studying, whether it's population genetics, whether you're looking at rare or common diseases. You need to narrow it down. Here, we're going to focus mainly on the part where we're identifying the variation in the first place. And you can expect that there's going to be some downstream analysis to do that additional filtering to find out what matters. And so the exercise of genomics, and particularly genomics in the cloud, is going to present a wide spectrum of challenges because it's a highly interdisciplinary activity that is going to bring together engineering, IT, life sciences, and although practitioners from those fields will be confronted with the jargon from the other field. If you have an engineering or IT background, you'll probably struggle to get to grips with genomics from a scientific point of view. There's a lot that goes into it that comes from biology, chemistry, physics, mathematics, statistics, and there's a lot of jargon that gets thrown around. Meanwhile, if you're coming from the life sciences, you're likely to encounter a level of complexity in computing concepts that you haven't had the formal training for. And so what Brian and I tried to do with the Genomics in the Cloud book is to lay out a practical roadmap for how to learn to do genomics in the cloud based on the current state of the art. And so in the presentation today, I'm going to talk about these key points. I'm not going to try to teach you all of the concepts because that's a 500-page book. And we don't have that kind of time right now. But hopefully you'll be able to get a sense of what you need to learn and what are the things that you're going to have to get to grips with. And you'll have a plan for actually tackling that concretely. So let's get started. Cloud basics. What are the key concepts that you need to know in order to work with the cloud for genomics? Well, the most foundational concept probably is virtualization. So this is the idea that you're going to use not regular computers in a sense, but virtual machines and containers. And what is that? Well, if you think about your basic computer, let's say the laptop you're using right now to view this, you have a physical machine, actual hardware that you can point at, and that's got some software layered on top of it. So you've got an operating system, which might be Linux or Mac or Windows, and then you'll have some libraries that provide some common functionality, and then the actual application that you want to run as an end user. In a genomics example, that would be GATK, which we'll talk about in a moment. So that's your basic machine. Now, when we're talking about virtual machines, that is a type of virtualization where we're abstracting away the basic infrastructure. You no longer have a specific physical machine, but there's kind of a layer of infrastructure. And on top of that, we can pretend that you have specific machines that behave like a real machine in your hands, but actually they're kind of a construct, a virtual machine. Containers, kind of a similar concept in terms of what it looks like to you as an end user, except it's a lighter way to do it. And you're not distinguishing several different machines, but you have one machine. And within that machine, you'll have different environments that are fully encapsulated and isolated from each other so that you can run things that are normally incompatible on the same machine and they won't interfere with each other. And in the cloud, actually, what you're going to use is a combination of both concepts, virtual machines and containers. So you'll have access to virtual machines on the cloud, and on these virtual machines, you'll use containers to run the software that you need to run. So that's a very important set of concepts to get to grips with. The good news is that in practice, it's not actually very difficult to use. And for example, Google Cloud makes it really easy to spin up a virtual machine. It's as easy as browsing a menu of basic configurations, picking one in the menu, and a few clicks later, you have a terminal and you can use the machine as if it was your laptop. Now, of course, when you're working on the cloud, there are going to be a few subtleties around how you interact with data. You no longer have data just on a local file system. The data is going to be stored in kind of one of two forms. One is object storage, which is here called cloud storage in this diagram. And you'll basically have your files in something called a bucket. And you can have any number of buckets, which mimic a file system, even though they're not quite the same thing. And when you have your files in a bucket, you can copy those files to the local storage of the virtual machine you're working on, do some work, then write the outputs either back to the same storage buckets or to a different storage bucket. You can manage buckets, including bucket permissions, share them with other people, you can lock them down, and so on. The other way that you might store your data on the cloud is in a data warehouse called BigQuery. And that's for tabular data of the type that you might normally have in spreadsheet formats. And so you'll have that tabular data in the BigQuery warehouse, and there's some utilities for interacting with that, making queries, extracting slices of the data that you want to analyze. Now, most of what we're talking about in this presentation involves files that are in object storage. And for that, you'll be using a utility called gsutil, which stands for Google Storage Utility, I believe. And that allows you to use command line commands to copy data, to move it around, do everything that you would normally do in a Linux file system, in a shell. So that's how you're going to interact with the data. The final point that I want to make is around working with containers. Now, containers, as I mentioned, the idea is to have an encapsulated set of software that includes the application you want to run as well as all of its dependencies, in a way that you can just pull an image, a container image that describes all of that software, pull it to your local computer, whether it's your laptop or a VM in the cloud, it works the same way. You can store that image, or you can run it to instantiate the actual container. And when you have the container up and running, there's a variety of ways in which you can use containers depending on what you're trying to do. And in the book, we go through some of the most common scenarios that apply to genomics. That really sums up the key things that you need to know in order to work with cloud infrastructure at a basic level. So far, hopefully that's not too bad, right? So now let's talk about the scientific side of things, and let's talk about the basics of working with GATK, which is the main genomic analysis toolkit that we're going to be using here. The Genome Analysis Toolkit, which is developed at the Broad Institute, is an open source toolkit. And it's really mainly focused on genomics, and specifically the variant calling part. But there's actually more than that. And at this point, there's about 200 tools in the GATK that allow you to do various data processing tasks and analysis tasks within the scope of genomics. Now, these individual tools can be chained together into workflows, and the GATK documentation includes descriptions of the recommended way to do this, which is what we call the best practices, GATK best practices. And there are GATK best practices workflows for all the major types of variant discovery analyses. And in the book, we describe in loving detail 3 of these workflows that are most commonly used genomic use cases. There's a few additional ones that are being developed that we will add to the repertoire as soon as they're fully mature. So, how do you actually run GATK? Well, running GATK itself is a matter of doing 2 things. First, you're going to pull the official GATK container, which is published by the GATK team, and then you can just run it. So you don't have to do any installation, you just spin up the GATK container. And you spin it up in interactive mode, and so once you're inside the container, you can just run GATK command lines. And here I'm showing you a command line for haplotype caller, which is the most commonly used variant caller in GATK. And you see it's a fairly simple command line where we're providing a reference genome, a file, what we call a BAM file, which is the file that contains the sequence data, and then the output is going to be a VCF file of variants, which describes the variation that the program will have found in that particular person's genome. And that's really all there is to running individual GATK tools on the cloud. So pretty straightforward. What's really nice as well is that we don't have to download the results in order to view what the program identified. One of the most common things to do once you have your results at a basic level is to look inside that VCF file and look at what variants were identified. Now, we're going to use the integrated genomics viewer desktop version here, which actually allows you to enable Google access. So you can set up your Google credentials in the IGV application. And once you do that, you can provide paths to where the files live in storage in the Google buckets and view the genomic information, both the sequence and the variants that have been identified, directly in IGV without having to download the file. Okay, so that's very convenient because we're talking about very large files. You don't want to spend the time to download all of that. Now, that's so far fairly simple, but the reality is that doing genomics at scale is not something you want to do command line by command line manually in a terminal. What you're going to need to do is use pipelines that automate the work. And here we're showing examples of three of the most commonly used GATK pipelines. And you can see that there are many steps. These steps we have here even abstract away some of the complexity of exactly which tool gets called how and at what point. So there's going to be some complexity to that. So the next thing you need to do is learn how to run genomic workflows at scale. So let's talk about pipelining. All right. The idea, as I mentioned, is to automate the analysis so that you don't have to be running individual command lines in the terminal. This is to save you from a lot of hassle, but also to be able to reproducibly apply the same processing and the same analysis to any number of samples. And so here we have a small subset of a very simple workflow script that will run the haplotype caller tool using a particular GATK command line and with placeholders for the input files that would get plugged in at the time that this gets run. So this is a very simple example. In a real genomic pipeline, you'll have various additional features. For example, you'll have instances of parallelization, where you're going to chunk up the genome, say, into different regions and process those regions independently. So in a real workflow, you'll actually have quite a bit of complexity. And here, they're not very readable, but the point is just to show you the idea that you have some overall organization into sections of the pipeline, and each of the sections that showed in the upper part of the graphic actually expands into a whole workflow that can have quite a bit of complexity in itself, as shown in the bottom part. So these workflows can get fairly complex, and it's worth spending some time learning the logic of how they are set up, and also what kind of workflow language they're written in and so on. And there's multiple workflow languages that exist. We use one that's called Workflow Description Language, pronounced WDL, WDL. But there are others like CWL, the Common Workflow Language, and Nextflow, which are also quite popular in genomics. Now, you have your genomic workflow that describes all the tasks, all the commands that need to be run, in what order and how. That's great. But so the next parts that you need to learn about is how to run those effectively on the cloud, and specifically how to use the available cloud resources effectively, both in terms of the time you spend and the money that you spend running these workflows. The really cool thing is that with the cloud, you're not limited to just one machine. So up to now, we've been talking kind of in the context of a single virtual machine where you're just running things on that one machine. With the cloud, you have access to a huge army of VMs. And so here we're showing how we have a workflow that we submit to a workflow execution system that's called Cromwell. Cromwell, it will interpret your workflow and generate individual tasks, submit that to a service in Google Cloud that's, here we're calling it Pipelines API. That was the original name. It's since been renamed to Life Sciences API. We're a little out of date, but Same concept, basically. Cromwell submits the job to the pipeline's API, which is then going to submit that task to a virtual machine. And what's really cool is that you don't need to orchestrate any of that. You don't need to manage a fleet of virtual machines. You just give the definitions of the task to the API, the Google Cloud Pipelines API, and it will orchestrate all the orchestration for you. So it's really cool because you can just go to the dashboard in the Google Cloud Console and just watch the VMs getting created. They're going to do the analysis work, and then they're just going to disappear, and you don't have to manage any of it. So that's a really cool thing. Now, there are a number of things that you can do to optimize your use of these cloud resources to save time and money. One thing you can do to save money is to use preemptible VM instances. These are virtual machines that are like regular machines from a technical standpoint, but basically, you agree that you will pay less money for them. The running cost is lower, but in exchange, the machine can be taken away from you at any point if there's a high level of utilization of that machine type on the cloud by other people. So if you're using a preemptible machine and somebody else who is willing to pay full price needs that machine type, Google Cloud will basically take away your machine and give it to them. That sounds like a raw deal, but it's actually financially really interesting because for short running jobs, the rate of preemptions is actually pretty low. And so even if you get preempted once or twice, you still end up paying less for the same run than you would if you were paying full price. So for example, in our graphic here, we have an example of a task that costs $25 to run. And I will say these costs per task are much higher than we normally see, but it's to give you something easy to work with. Let's say it costs $25 to run full price. Now we've got another one that's analogous, but that you send to a preemptible machine. Let's say it gets preempted some way halfway through the run, and you pay $2 for the runtime that you did use. The system will restart your job with a new preemptible machine. When that runs to completion, and that costs, let's say, about $5, because typically right now the list price is about 20% for the preemptibles versus the regular VMs. You still in total only paid $7 compared to $25 for the same amount of work. So if you're willing to have the chance of being preempted a few times, you can actually save a lot of money on your total pipeline costs. And for the whole genome pipeline that we run in production at the Broad, we use preemptible instances pretty much everywhere. And it does represent a huge savings. Now, another way to save time and money is to use the call caching feature of the Cromwell execution engine. And this is basically a system that's also sometimes called job avoidance that allows you to repeat a pipeline, to repeat a workflow that was interrupted and resume at point of failure. So instead of rerunning everything, the system will automatically recognize what has already been run successfully and only restart the jobs starting at the point of failure. This can also be useful if you're testing a pipeline and you're changing steps that are halfway through the pipeline. You don't have to pay attention to where you restart your test run. You can just rerun the whole thing and the system will automatically restart from the point where something has changed. And so that's a feature of the Cromwell workflow engine that's very convenient. Now, to take advantage of that, you need to have a Cromwell server that you're managing. There's a little bit of operational overhead to that because you have to manage a server, you have to manage a database. It's not quite as trivial as just running a program in a kind of one-off way. So if you don't want to manage your own server, you have the option of using a Cromwell server that we manage that's in a platform called Terra, which I'll talk a little bit more about in a minute. And that's a Cromwell server that we manage, that we make available to the research community for free, and that anyone can use. And the idea is that you have your workflows and your data loaded in the cloud, specified in a workspace on the Terra platform. Then you can dispatch that to the Cromwell server in Terra. And that will orchestrate everything, the interaction with Google Cloud and the execution of your jobs for you. And so it's a very convenient way to run your pipelines if you don't want to manage those resources yourself. So it's really nice because you can send off any number of samples, and there's some functionality to be able to manage large data sets and just run a workflow on 10,000 samples. You click a button and then you sit back and relax while the system does everything for you. Now, Terra is not just a service for running workflows. There's actually quite a bit more to that. I'm not going to go into the full details, but basically it's a secure open platform for data access and analysis that is developed and operated by the Broad Institute in collaboration with Verily. And as you can see on this diagram, there's the workflow management capability that I just talked about. But there's also some capabilities for interactive analysis. So you can spin up Jupyter notebooks on a customizable computing environment. We're also working on a RStudio implementation. So you can do interactive analysis, interact with the data, and run code without having to download any of your data out of the cloud. There's a library of data sets that is accessible through Terra. Most of these data sets are that are hosted by other organizations, but they are connected to the system so that you can access the data in these data sets and these projects. And you can even combine data from different projects. For those data sets that have some access control restrictions, there's a system for gaining access to the data sets with appropriate credentials and so on. And so the goal here is to make it much easier to work with different data sets, do federated data analysis, and generally simplify your life in terms of how you access and work with a variety of data sets. One of the things that is really near and dear to my heart is the workspaces that are made available in Terra to make it easier for people to use tools like GATK. So the GATK team and the support team provide workspaces in which all the GATK best practices pipelines are fully set up. All the code is in there. There's example data and everything is configured. So you can just go in, you clone the workspace, and you can run it out of the box and it'll run right away. And you can also upload your own data and then run the pipelines on your data with very little setup work. So I think that's a really neat way to make these pipelines available to the research community. And finally, and this is kind of an extension of this concept, is that researchers can make workspaces public that contain all the code and data and artifacts to reproduce the work that they publish in their papers. So for example, here we're showing an example of a preprint in bioRxiv by a team at the Klarman Cell Observatory at the Broad, which is this workspace provides access to notebooks that have all the code for reproducing the computational analysis in this single cell transcriptomic study. And so I think this represents the future of scientific publishing, this idea of having something that's not just a method supplement with some description of the method and maybe if you're lucky a pointer to GitHub, but actually executable artifacts that allow anyone to reproduce the work that was done in the paper and then apply those same analyses to their own work. And I'm really hopeful that this is something that will catch on and will facilitate sharing and collaboration and general computational reproducibility of published research. So there's a lot more to learn about Terra than I can talk about in the time we have here. I recommend you check out the Terra homepage itself. The user guide and support has an extensive documentation and knowledge base. And if you want to get started, your interest is piqued, I really recommend looking at the Intro to Terra video playlist that is published on YouTube. That is a series of very short videos to give you an overview of all the things you can do with Terra and get started. Or, of course, you can get the book, Genomics in the Cloud, published with O'Reilly, that Brian and I co-authored, which is kind of the handheld roadmap to doing genomics in the cloud with all of the components that I've talked about today. So we hope this will be helpful in getting you to work productively on the cloud, and we'd love to hear from you about the webinar today, about the book, and your journey to the cloud. So, Joe, thank you very much. I'll pass it back to you.
Thank you so much, Geraldine. That was a great presentation. I'm sure folks learned a lot from that. We're going to move on to some question and answer now with Geraldine and Brian. So, for the first question, how much experience with cloud do I need for this book? And how about general computing and coding experience?
Yeah, I think that's a really good question. Geraldine and I, when we wrote the book, we really tried to make it as accessible as possible. So we tried to make it welcoming to newcomers to the cloud. You don't need cloud experience, so you can jump into this book and learn about the cloud through this book. Something that is useful for experience here is a little bit of shell experience, a little bit of experience with the Linux shell. So if you know what commands like ls and cat do, you're in a good state. If not, like we actually in the preface of the book have links out to really awesome tutorials that will bring you up to speed. Likewise, on the coding side, we have examples of R and Python code later on in the book. And those are really there to give you a taste of what's possible in the Terra platform. So we hope it's a good jumping off point. You don't need to be an expert on R or Python code to appreciate what those examples are trying to show. Hopefully they're a good jumping off point for future investigations on your side and future learning.
Okay, great. Thanks. Does the book cover multiple clouds?
So that one, for now, the book is entirely set in Google Cloud, and that's really because that's what we use at the Broad. And as they say, you gotta write what you know. But we did try to put a lot of thinking into writing it in a way that would be applicable beyond just Google Cloud, because a lot of the concepts are common across the clouds, and the important pieces that you learn are about the same, right, Brian?
Yeah, I think that's the key point here is it's the key concepts about what are VMs, what are containers, what are workflows that we're trying to explore in the book. And these are good sort of foundational background pieces of information that are highly portable from cloud to cloud. So there are specifics, things like preemptible instances on Google are a little bit different than spot instances on Amazon. But the basic sort of foundational knowledge is the same, and how those particular tools can be used to kind of lower your cost of analysis on the cloud. That's a common sort of thread that exists between the various cloud environments. The other thing that I think is really useful to point out here too is, in terms of Cromwell as the workflow engine behind the scenes, doing those sort of workflow launching in Google Cloud and whatnot, that's actually a very portable piece of software. So the Cromwell workflow engine has plugins for Google, of course, but also Azure and Alibaba, and there's an AWS batch backend as well. It even supports some HPC environments, so you could really use it to kind of span your work across multiple clouds. So again, the knowledge that you pick up in the book, definitely the themes and the background is going to be useful as you transition from environment to environment.
Great, fantastic. What about Terra? Is it Google only, or does it support other clouds?
Yeah. So Terra right now, when you log into Terra, you're logging into an infrastructure that's on Google Cloud. So it is a Google Cloud based platform. But the Terra team, is working on exploring support of other cloud environments. And the ultimate vision here is that what you want to be able to do is log into Terra and use data from a variety of sources and a variety of different cloud environments and regions and be able to compute on it. So that's the long-term vision here, and it's something that the team is actively working towards. I would say like probably a good way to keep tabs on the development of multi-cloud support in Terra is to look at the Terra blog and subscribe to that. And there's going to be regular updates over time about how Terra embraces sort of multiple cloud environments.
Got it. Why should I use cloud since I cost money when my university has an HPC cluster that I can use for free? And can I use what I learned in this book there?
Well, that's a great question, and honestly, the first half of the book, which is really about GATK and the best practice of workflows and all of that, that will be broadly applicable whether you're working on a HPC or on cloud, give or take a few details. So that's I would say that is completely cross-platform. It's really the second half that is more specific to the cloud. Um, but I would say that we do hear a lot about local HPC being available for free. So why wouldn't you use that instead of paying for cloud on top? The thing is, though, that your HPC is not actually free, and somebody somewhere is paying for it. Usually, it's taken from grant overhead, so you don't really see it, but you also don't have the option to not pay for it. It's more like it's prepaid rather than free. So I guess the question for a lot of people then is like, why would I pay two times for doing my analysis instead of just the one, right?
Yeah, I think just to kind of echo that, there's an argument here for efficiency, right? So I remember as a graduate student patiently waiting in the SunGrid engine queue, hoping my jobs would schedule out over the weekend. Of course, they would always fail over the weekend. When I would come back Monday, I have to resubmit them when everyone's adding their stuff to the queue. So those sorts of painful experiences of using shared resources, are quite common, right? What's wonderful about the cloud is this sort of capacity. So if I'm a researcher and I really want to do my analysis right now, the cloud has capacity. It lets me much more quickly turn around my analysis, get an answer, refine it, do the work again, converge on an answer. That turnaround is worth quite a lot in my book. So the ability to have a very efficient, available environment is one of the key aspects, the key differentiators for HPC versus cloud, I would say.
Great. So, big data question here. What's the most data in terms of genomes or samples that you've pushed through Terra?
Ah, okay. That I think that's a good question. I would like to compare and contrast that. So I would say a few years ago, or maybe several years at this point, the largest project I had worked on was about 6,000 whole human genomes spread across a few different cloud and HPC environments. And this was work done in the PCOG project. So at the time, that was a big project, and we did a sort of home built software for the clouds to do all the sort of provisioning and infrastructure necessary to run analysis on those. And that project ended up taking literally, a couple years because of the sort of overhead of building everything and building all the infrastructure. Fast forward to today, and I'm personally working on projects where we're talking about 10, 20, 25, 30,000 samples being pushed through Terra and the Google Cloud platform. And from my experience there, the scalability and being able to rely on these services is such a night and day difference between the work of PCOG just a few years ago. So I think that's really exciting. Looking forward, looking forward a few years, we have projects that will have 300,000 samples, 400,000 samples. So I think it's really exciting to see the system scale now, and also to see what it will be able to scale to in the future.
Yeah, and like we should mention that the GATK team right now is working on scaling the joint calling algorithms to work on, I think, 1 million genomes, which is a little mind-boggling if you think about the size of the VCF file. Except, of course, they're not using VCF files anymore. At that point, you need to use different solutions, which is part of the exploration that's going on right now.
Right. That's been very interesting. I've been involved in that project with the Brody. So very interesting. Question from somebody. I don't like GUIs. I feel like they just get in the way. Can I use scripts to use Terra capabilities programmatically?
Yes, you can. So the cool thing about Terra, and also the Google Cloud platform as well, is that what you see in the GUI is actually backed by an API. So if you can see it and click on it, you can use an API to access it. And so that's really powerful because you can script on top of these APIs using in Terra, there's a library called Fizz, F-I-S-S. That's a Python library for the Terra API. And what that lets you do is it lets you do automation, like creating workspaces, loading it up with your data, even triggering workflows. So I think that's a really powerful aspect of the Google Cloud, but also, Terra is the fact that it does provide those APIs so you can mix and match whenever it's appropriate. Sometimes I use the GUI when it makes sense, and other times I script on top of it.
Great, great. Another question here. Can I use RStudio or other workflow languages or environments in Terra?
Yeah, so at the moment, Terra has Jupyter Notebook support, as you probably see in the book and the presentation that Geraldine gave, and that allows you to support multiple coding languages. So you can embed Python or R code directly within those notebooks, and so that's something that you can use today. Something on the horizon that's being worked on that I've seen awesome demos for is expanding support of notebook-like systems. So RStudio is being added, and as well as Galaxy, which is a sort of workflow engine and visualizer. Those are both being added as sort of interactive analysis components within Terra. And you'll see additional things like this added over time. And I'm personally very excited to see those. I know researchers find them extremely useful. The other thing I want to just say there is the Cromwell that Terra is using. That supports Whittle, but if you use the Cromwell standalone workflow engine, like I mentioned earlier, that actually supports Whittle plus CWL. So you see multiple workflow language support in that component. And so that's something that over time, I think there will be more options as those components of Terra evolve.
Great, great. One last question. What's the best way to get help with the book?
Yeah, so I think Geraldine mentioned in the slides the support.terra.bio. That's a great resource, right? So there's a lot of tutorials and things like that to enrich the content of the book, and also ways to ask questions. So that's a really helpful resource, and we've worked a ton with that group. They're aware of the book and have given feedback on the book. So from a support perspective, that's a great resource. The other thing I would say too is, just as a quick plug, if you find something off in the book, or something isn't quite working the way that you'd expect it, and you find basically a bug in the book, we'd ask you to submit through the errata page on the O'Reilly website. And that way we can just fix it and make sure that the book is kept up to date.
Yeah, and we'd really love to get your feedback in general on how useful you find it, and what parts you struggle with, and so on. I'm I'm kind of active on Twitter, so don't hesitate to reach out to me there if you want to talk. And you can also find me on LinkedIn and in a few Slack workspaces, including Open Whittle. And I always really enjoy having interactions with people who are using the software. Please tell us how you feel, and we'll do our best to help you.
That's fantastic. Well, Geraldine and Brian, thank you so much for this really informative session. I'll pass it over to Luis to close things out.
Thank you for tuning in. Thank you, Brian. Thank you, Geraldine. Thank you, Joe. We are going to go ahead and follow up with all of you who joined live. And if you have any questions, please contact your Google representative. Please stay safe, everybody. Thanks again.